Hello everyone. We have discussed in our previous lecture about aligning two sequences and assigning a score to each of these matches, mismatches, and gaps. Now, assigning a score follows certain scoring schemes. For this, you have different substitution matrices for nucleotide alignments and different substitution matrices for protein alignments. In this lecture, we will discuss about substitution matrices for nucleotide alignments. Nucleotide substitution matrices are two different kinds of matrices. One, the identity matrix, and the other one, which we are going to discuss, is the transition or the transversion matrix. In the identity matrix, a matrix is scored or a substitution is scored by a match or a mismatch. That's it. In an identity matrix, once again, I repeat that the scoring is done for a particular match or a mismatch. Now let's see clearly about this. See, here in this particular substitution matrix, you have A matching with A and it is given a +1. Any mismatch is given a -1. And similarly, T matching with T will be given a +1 and G matching with G will be given a +1 and C matching with C will be given a +1. That means all diagonal elements will be a +1 and all the upper triangle, this one, and the lower triangle, this one here, what you have, right? Both of them are having negative scores. And this is what is an identity matrix. Sometimes mismatches are also given a score of 0. Now, we'll go to the transversion or transition matrices. The transversion or transition matrices, if you're talking about, transition is between the same group. For example, you're talking about between purines. Both of them are purines, and if the substitution is from A to G or from G to A, then it is called as a transition. And similarly, if it is from C to T or from T to C, it is called as a transition. And anything other than that is called as a transversion. Means from a purine to a pyrimidine or a pyrimidine to a purine, they are called as transversions. Now look at this. In this transversion or transition matrices, if you remember, we were talking about certain similarities. However, in that case, when we talked about similarities, we talked about that the word similarity is always used only for protein sequences or amino acid alignments. Here, the only difference here is the transversions here, which you have A to G, will not be having that a negative score as that of any other mismatch. Means the score given to a transversion is slightly lower, means it's penalized lower than the one that is given for a mismatch. However, the score here is negative. Remember, when I was talking about similarity, the score is never negative. It is greater than 0. That's what we have discussed, and it's used for proteins. So similarity, anyways, is used only for protein sequences. But just because A and G belong to the same group and it is a transition, transitions are penalized lower than the transversions. And that's why here, all the transitions that you see in this table are penalized lower than the other mismatches. Also, we'll discuss which matrix to use when. The identity matrix is used when comparing highly similar or nearly identical sequences. And this is generally used for PCR primo matching or exact match checks. And this is also used to align reads to the reference in high fidelity short read sequencing that is used in NGS. We'll come to that later on in the later lectures. This should be avoided for evolutionary divergence or mutations in the sequences that you are comparing with. And when is transversion or transition matrix used? The transition or the transversion matrix is used when you are performing evolutionary comparisons between DNA sequences, or you're modeling mutation bias. That transitions are more frequent than transversions. And when you're working with phylogenetics, especially the mitochondrial or the viral nucleotide or DNA sequences. Thank you.